once we have an understanding of how an action potential is generated along the axon of a neuron, then we can understand how that signal is passed on to our neighboring neuron. So remember that along the axon, we had this action potential generated, which caused sodium channels and potassium channels to open up, and it caused the inflow and outflow of those ions. Now we've reached the end of the axon, known as the axon terminal. Now that action potential, when it reaches the end of the axon, it causes the voltage-gated calcium 2+ channels to open. And since there's a higher concentration of calcium 2+ outside of the cell, calcium will run into the axon terminal. When it runs into the axon terminal, this causes the vesicles that carry the neurotransmitter to bind with that cell membrane. So once again, what happens is that action potential propagates to the end of that axon terminal, so we're right over here. And when that action potential reaches the end of that axon, it causes the calcium channels to open, and calcium will rush into the axon terminal, causing the vesicles to bind with the cell membrane. Now, these vesicles are carrying some sort of a neurotransmitter based on the signal that this neuron initially received. So this neuron that is passing on the signal to the second neuron is referred to as the presynaptic neuron. Pre meaning before or first. So this is the presynaptic neuron, and it's passing on the signal to the second neuron, known as the postsynaptic neuron. So we're going from pre to post. Now, when that vesicle binds to that cell membrane, it causes the neurotransmitters inside those vesicles to be passed on to that second neuron through exocytosis. So these neurotransmitters will leave the vesicle through exocytosis, and they will now enter the synaptic cleft. The synaptic cleft is essentially the space in between the axon terminal of the presynaptic neuron and the dendritic spine of the postsynaptic neuron. So remember that neurons, they receive signals through their dendrites, and then they pass it on to the axon hillock. So right now we're at the dendritic spine, so the dendrites are receiving that signal, and that signal is passed on through these neurotransmitters. Now, on the surface of the dendritic spine, or the dendrites, we have these receptors. So these are neurotransmitter receptors, which will receive the signal of the neurotransmitter. So the neurotransmitter binds to that receptor, and it causes some sort of a reaction in that second neuron. So once again, to quickly sum up what happens, we have the action potential coming down the axon. When it reaches the axon terminal, the calcium 2+ channels open up, and since there's a higher concentration of calcium 2+ outside, this causes the calcium to enter the axon terminal, which results in the vesicles fusing to that cell membrane, and the neurotransmitter is released via exocytosis into the synaptic cleft, where it will then bind to neurotransmitter receptors of the postsynaptic neuron on its dendritic spine. Now, when that neurotransmitter binds to the receptors of that postsynaptic neuron, it can have excitatory or inhibitory effects. Now remember that whenever we receive some sort of a signal from a neighboring neuron, that those signals that we receive are summated at the axon hillock. And remember that the axon hillock is the location that basically connects the axon to the cell body of the neuron. So the axon hillock is where the signals will summate, and the neuron will determine whether an action potential will occur, and if some sort of reaction occurs, will it be excitatory or will it be inhibitory? Now, let's take a look back at our resting cell. So we know that the resting potential of a neuron is -70 millivolts, and that resting potential is established with the help of our sodium-potassium ATPase pump, which is pumping 3 sodiums out and bringing in 2 K+, and creating that net change of +1. So the outside of the cell is actually more positive, and the inside is more negative. So that creates that -70 millivolt voltage, which means that the inside is 70 times more negative in comparison to the outside. Now, if we receive some sort of an excitatory effect to our postsynaptic neuron, that means that we have an action potential occurring. So that means that the signal is strong enough and we surpass the threshold and we have this depolarization event. Now, this depolarization event is defined as sodium rushing in, because remember, during our depolarization event, the sodium channels will open up, and since there's a very high concentration of sodium on the outside of the cell, the sodium is going to rush in and move along its concentration gradient. And when it moves in, we see a lot of positive charge in here. So the inside of the cell becomes more positive. So we say it becomes approximately positive 40 millivolts, which now means that the inside has become 40 times more positive in comparison to the outside. So that's an excitatory effect. And in an excitatory effect, we see depolarization and we see sodium coming in. But how would we see an inhibitory effect? An inhibitory effect is essentially making the neuron hyperpolarized. Remember that the hyperpolarized event is defined over here. How do we become hyperpolarized? Now, if we remember back to how an action potential occurs, we can recall that during repolarization, we see the potassiums moving out. The potassiums move out because we can see over here that we have a lot of positive charge when those sodiums come in. When those sodiums are coming in and the cell is depolarizing, we have a lot of positive charge, and the potassiums will then want to leave. When the potassium leaves, that's our repolarization event. And the potassium channels, they're very slow to close, so we have too much potassium leaving. Too much potassium leaving causes a hyperpolarization event, and during the hyperpolarization event, we have this refractory period. And the refractory period is split in half between the absolute refractory period and the relative refractory period. Now, if we enter the refractory period and we're hyperpolarized, it becomes more difficult for an action potential to occur because we're not at our resting state. If we want an action potential to occur, we must be at our resting state. Now, if we want an inhibitory effect in our postsynaptic neuron, what's going to happen is that we're going to become hyperpolarized. That means that potassium must exit. So once again, when we have an excitatory effect, we become depolarized, so sodium will come in an excitatory event, but in an inhibitory event, we will see potassium leaving and we become hyperpolarized, because when you're hyperpolarized, it's more difficult for the neuron to elicit some sort of an action potential. And that is essentially signal transmission between two neighboring neurons.